Hello, my dear students. Welcome you all to Sri Jaindra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the last video, we have started. Actually, we were concluding with the excretory system. We were discussing about the excretory system of humans. We have discussed the process of glomerular filtration, different terminologies associated with glomerular filtration, that is net filtration pressure, renal plasma flow, filtration fraction, GFR. We also studied about the factors affecting the glomerular filtration rate. We were on selective reabsorption. We discussed about the reabsorption at the level of proximal convoluted tubule. And now we are going to talk about the distal convoluted tubule. Distal convoluted tubule, it is an area for conditional reabsorption. Very important. Conditional reabsorption. In the previous video, we studied about the renin-angiotensin-aldosterone system. Conditional reabsorption. Okay. So it is a area, it is a place for conditional reabsorption for water, Na plus and Ca2 plus. The conditional reabsorption or the facultative reabsorption of water with the help of ADH, Na plus with the help of aldosterone, and calcium ions with the help of parathalamone. When the level of plasma water falls, mean a condition of hypovolemia, the posterior pituitary, means the neurohypophysis, releases the antidiuretic hormone, that is ADH, which increases the permeability of distal convoluted tubule. The secretion of ADH, antidiuretic hormone, basically it increases the permeability of distal convoluted tubule and the collecting duct to water. So permeability towards water is basically altered for distal convoluted tubule and the collecting duct. Understood? Water is reabsorbed from the filtrate by osmosis, and the reduced amount of concentrated urine is produced. Mean hypertonic urine is produced. Okay. At the level of DCT, the urine is hypotonic. Okay. Here, 13% of water is reabsorbed by facultative reabsorption. Very important. Okay. So, ADH, antidiuretic hormone, also known as vasopressin, it is basically synthesized by hypothalamus and released by posterior pituitary. It is secreted in response to high blood osmolarity. High blood osmolarity. Okay. The normal levels of osmolarity at the level of DCT is 300 milliosmoles per liter. So when the level of plasma water falls, okay. So level of plasma water falls means the water is less in the filtrate. Okay. So the water level is below, the urine is concentrated. The posterior pituitary releases the antidiuretic hormone, which increases the permeability of distal convoluted tubule and collecting duct to water. Water is reabsorbed from the filtrate by osmosis, and reduced amount of concentrated urine is produced. Here 13% of water is reabsorbed by facultative reabsorption. Okay. So if the osmolarity is 300 plus, then the ADH gets activated. Okay. High osmolarity is detected by osmoreceptors in hypothalamus. So osmoreceptors, they are present in hypothalamus. Are present in hypothalamus. So these are osmoreceptors, basically judge, they analyze the osmotic pressure, the osmolarity of the blood, and that is why after the activation of these osmoreceptors, they trigger the hypothalamus to release, to directly form the hormones, okay, which directly store in the posterior part of the pituitary, and they are secreted. Okay. There is a thirst center in hypothalamus which is stimulated and intake of water occurs. Very important. So the activation of hypothalamus basically activates the thirst center. Stimulated in hypothalamus. Okay. Now, the distal convoluted tubule and the collecting duct actively reabsorb sodium from the filtrate under the influence of adrenal hormone aldosterone. Okay, so the water reabsorption takes place through facultative reabsorption, okay, by osmosis, and the active transport of sodium ions from the filtrate under the influence of adrenal hormone aldosterone takes place, which makes their walls permeable to the ions. The reabsorption of sodium ions brings about the uptake of an osmotically equivalent amount of water, but the duct of Bellini is relatively impermeable to water. Very important. The duct of Bellini is impermeable to water. Bicarbonate ions are also reabsorbed in DCT. Bicarbonate ions are also reabsorbed in DCT. So ADH basically makes DCT and the collecting duct permeable to water, which leads to the formation of concentrated urine. If osmolarity becomes less than 300, then further ADH secretion is stopped. Okay. So the ADH secretion, it basically triggers when the osmolarity is more than 300. Okay, when if the hyperconcentrated urine is there. Okay, so this is the function of ADH on DCT and collecting duct. Along with the ADH secretion, okay, the aldosterone secretion for sodium ions reabsorption, okay, also takes place. The duct of Bellini is impermeable to water, and the bicarbonate ions are also reabsorbed in DCT, that is distal convoluted tubule. So the distal convoluted tubule, it is the center for conditional reabsorption for water, Na plus and Ca2 plus. Understood? I hope you got it. Okay. Tubular secretion. In tubular secretion, before moving to tubular secretion, I just want you to, look some more points regarding the descending limb and the ascending limb. As we have already studied that the descending limb is permeable to water only. It is only responsible for reabsorption of water. And the ascending limb is permeable to ions only, no water. Okay. Collecting duct is basically for conditional reabsorption of H2O, calcium and Na plus as we have discussed. It shows permeability for urea. It shows permeability for urea. So please remember the collecting duct. This is the point I actually want to mark it. Okay. The collecting duct shows conditional reabsorption of water, Ca2 plus, Na plus, and it shows permeability for urea, the collecting duct. Okay, this is for collecting duct. Understood? The glomerular filtrate and the in the PCT are isotonic to plasma and DCT is hypotonic to plasma. And in the hairpin loop, it is hypertonic to plasma. So please remember these points. Tubular secretion. Certain substances like creatinine, hippuric acid, and foreign substances like pigments, drugs induced penicillin are actively secreted into the filtrate in the proximal convoluted tubule from the interstitial fluid. Hydrogen ions and ammonia are also secreted in the proximal convoluted tubule. Hydrogen ions and the ammonia in the form of ammonium ion NH4 plus are also secreted in the PCT proximal convoluted tubule. Potassium, hydrogen, and the other ions are also secreted by active transport into the filtrate in the distal convoluted tubule. Okay, so there are certain substances which are extracted from peritubular capillaries and added to the filtrate, like K plus, H plus, NH4 plus, creatinine, hippuric acid, drugs, PCT, DCT, and collecting duct are concerned with secretion. They all are concerned with secretion. Like PCT, it helps in the secretion of creatinine, hippuric acid, foreign substances, okay, which are actively secreted into the filtrate, the PCT, from the interstitial peritubular capillaries. Hydrogen ions and ammonia are also secreted in the proximal convoluted tubule. Okay. Now, urea enters the filtrate by diffusion in the thin region of ascending limb of loop of Henle. Okay, so the thin region of the ascending limb of loops of Henle, okay, is basically permeable for urea. The urea enters the filtrate by diffusion in the thin region of the ascending limb of loop of Henle. The removal of these substances in the PCT and the DCT helps to maintain the pH of the blood. So basically the removal of H plus ions, okay, in the proximal convoluted tubule and distal convoluted tubule helps to maintain the pH of the blood between 6 to 8. Okay, any variation from this range is dangerous. It can lead to acidosis or alkalosis. More amount of removal of H plus can lead to alkanosis. More amount of removal of bicarbonate ions or ammonium ions can lead to acidosis. Okay. So maintenance of pH of blood is very important, which is actually done by these processes, tubular secretion. Tubular secretion probably plays only a minor role in the function of human kidneys. Very important. Play only the minor role, but exactly speaking, this role is whatever said to be minor, but actually it is very important, right? It is very, very important. But in animals such as marine fish and desert amphibians, which lack glomeruli and Bowman's capsule, tubular secretion is the only mode of excretion. Very important. So the ones who are not having Bowman's capsule and they don't have glomeruli, they are only, in support of this tubular secretion, which act as a mode of excretion. When the blood pressure and consequently the filtration pressure drop below a certain level, filtration stops and urine is formed by tubular secretion only. When the blood pressure and subsequently the filtration pressure, it basically gets stopped, then the filtration gets stopped, okay, and the urine is formed by tubular secretion only. Sorry. Now we are going to talk about the diuretic substances. What are these diuretic substances and what are their functions? Okay. The diuretic substances are the substances like you basically, you know, we all are in a habit of taking these diuretics in day-to-day life. Okay. So diuretic simply means what? Diuretics, antidiuretics means what? Normally, the amount of urine formed, it depends on the intake of water. The amount of urine formed, it depends upon the intake of water, dietary constituents, environmental temperature, mental and physiological states of the person. However, there are some substances which increase the volume of urine to be excreted. These substances are called diuretic. So diuretics are ones who are going to basically increase the volume of the urine. Okay, these are diuretics. Okay, example tea, coffee, alcohol. Okay, so antidiuretics basically they are going to maintain the volume of urine. If the urine is excreted and if it is hypertonic, then more and more amount of water will be lost. Okay. So to basically save that amount of water, ADH antidiuretic hormone is secreted, which helps in this function. Okay. The ADH it basically conserves the water. It conserves the amount of water. Okay. Understood? The materials which are transferred in traces like glucose, amino acids, proteins, albumin, vitamins, hormones, Na plus, K plus, Mg2 plus, Ca2 plus, bicarbonate ions, creatinine, ketone bodies, urea, uric acid, they all are basically the primary filters of Bowman's capsule. The process is involved in glomerular filtration, okay, and the mechanism is ultrafiltration, okay. Now, glucose, amino acids, hormones, vitamins, Na plus, K plus, Mg2 plus, and Ca2 plus. They are after getting reabsorbed in Bowman's capsule. If some of the amount isn't still in the glomerular filtrate, then they are reabsorbed by proximal convoluted tubule or PCT. The process involved is reabsorption, and the mechanism is active transport. So active transport of all these substances. Cl minus ions also PCT, reabsorption, passive transport, very important. So Na plus it enters the PCT, the brush border site with the help of active transport, and the Cl minus it basically moves through passive transport. Water, proximal convertible, reabsorption, osmosis. Okay. Urea, proximal convertible, reabsorb in proximal convertible, reabsorption, process, diffusion. Okay. H2O, narrow region of descending loop of Henle, reabsorption. The mode, the mechanism involved is osmosis. Okay. So water in the descending limb of loops of Henle, it is reabsorbed with the help of osmosis. With the help of osmosis. Sodium ions, potassium ions, magnesium ion, calcium ion, they all are reabsorbed in PCT through the process of active transport. Chloride ions are reabsorbed at the level of PCT with the help of passive transport. Water in PCT is reabsorbed with the help of osmosis. Okay. In the loop of Henle, descending limb, then also it is reabsorbed with the help of osmosis only. Sodium, potassium, magnesium, calcium, chloride in the thin ascending loop. Okay. Diffusion. They enters through diffusion. Inorganic ions as above, white part of the ascending limb of La, reabsorption is their active transport. Okay. Water, distal convoluted tubule, facultative reabsorption, collecting tubule, collecting duct, reabsorption with the ADH help, osmosis. Okay. Na plus, distal convoluted tubule, collecting duct, collecting tubule with the help of aldosterone, reabsorption with the help of aldosterone, active transport. Very important. Active transport. Okay. So urea, the last part of the collecting duct, urea, reabsorption with aldosterone help, reabsorption, secretion, diffusion. Okay. Creatine, hippuric acid, foreign substances, PCT, reabsorption with aldosterone help, reabsorption, secretion, active transport. Okay. K plus H plus distal convoluted tubule, reabsorption with aldosterone help, active transport. Okay. So K plus H plus active transport. Reabsorption is done with the help of aldosterone at DCT. Creatinine, hippuric acid, foreign substances. Okay, they are reabsorbed with the help of aldosterone. Okay, also help in secretion with the help of active transport in PCT. Okay, urea in the last part of the collecting duct, diffusion. Na plus in the distal convoluted tubule, collecting duct and collecting tubule, reabsorption with the aldosterone, active transport. Water, distal convoluted tubule, osmosis. Okay, the water is basically concerned with osmosis only every time. Okay, while the active transport is basically involved for the we can say for inorganic ions, Okay, sodium ions, okay, whether it is at PCT, whether it is at DCT, okay, but very important, these ions at the level of thin ascending limb of loop of La, then they are basically reabsorbed through diffusion. It is not of active transport. Okay, so ammonia is basically ammonia. It is not mentioned here. Ammonia is also reabsorbed at the level of distal convoluted tubule. Okay, the reabsorption with the help of aldosterone. Okay, diffusion. Okay, so ammonia, distal convoluted tubule. Okay, it also helps in the reabsorption with the help of aldosterone secretion. Diffusion. The mechanism is diffusion. Okay, urea. Then urea, ascending limb of loops of Henle, thin ascending limb. It is also through diffusion. Okay, diffusion. So this is the summary of all the things, all the events which occurred in nephron. Okay. Now I'm going to talk about the hormonal control of renal function. Okay. Control of antidiuretic hormone ADH produced in the hypothalamus of the brain and released into the bloodstream from the pituitary gland. Enhances fluid retention, conservation of fluid. Okay. By making the kidneys reabsorb more water. The release of ADH is triggered when the osmoreceptors in the hypothalamus detect an increase in osmolarity of the blood above the set point of 300 milliosmoles per liter. In this situation, the osmoreceptor cells also promote thirst. Drinking reduces the osmolarity of the blood, which inhibits the secretion of ADH and thereby completing the feedback circuit. Okay. So, when we talk about the role of ADH antidiuretic hormone in maintaining the volume of urine, it acts this way. Okay. Then the RAS system we have already discussed, that the control of juxtaglomerular apparatus, low blood pressure triggers the RAS system. Low blood pressure. Okay. Low blood pressure basically triggers the RAS system. Okay. Then we have atrial natriuretic peptide also, ANF, or atrial natriuretic factor. Okay. ANF also helps to control, okay, the blood pressure. It is secreted by the wall of atria. That is heart. It is secreted in response to high blood pressure. The triggering response is high blood pressure. Okay. High blood pressure, high sodium ions concentration, high blood volume. Okay. It opposes the RAS and ADH. It basically opposes the RAS and ADH. Regulation of urine formation by the feedback mechanism. This is what we can say thirst center. Okay, the osmoreceptors they activate thirst center. The thirst center it basically helps the person to drink more and more amount of water, which indirectly decreases the blood osmolarity. Okay, the hypothalamus releases ADH. ADH basically stores in the posterior port of the pituitary and is secreted in the blood. Okay, This vessel pressure basically constricts the vessels. It maintains the volume of the urine. Okay, it helps in the reabsorption of water along with the reabsorption of sodium ions. Okay, so higher blood volume or pressure, higher blood volume or pressure. It further activates the higher blood volume or the pressure. It further activates the aldosterone. Okay, so the factors like the RAS system and the secretion of ADH, they are triggered by the same thing. Okay, low blood pressure. Okay, so it is triggered by low BP. It is also triggered by high osmolarity. Okay, hormones which help in affecting the kidney functions like parathormone. Hormone increases the blood calcium levels, hypercalcemia, and decreases the phosphate level, blood phosphate level, hypophosphatemia. Accordingly, it increases the absorption of calcium, okay, increases the excretion of phosphate, okay, PO4 3 minus. So parathormone, it increases the blood calcium level, hypercalcemia, and decreases the blood phosphate level, hypophosphatemia. Okay, accordingly, it increases the absorption of calcium and increases the excretion of phosphate ion. Thyrocalcitonin, it increases the excretion of Ca2 plus in the kidney. Thyrocalcitonin, it increases the excretion of Ca2 plus in the kidney. Parathormone, it works oppositively. It increases the blood calcium level. It decreases the excretion of calcium. It increases the you know excretion of phosphate. Prostaglandins, the renal pyramids produce fatty acids and prostaglandins PG, which participates in blood pressure regulation. Erythropoietin is secreted by the juxtaglomerular apparatus and plays an important role in erythropoiesis, that is, production of RBCs at high altitude regions or where the low oxygen tension is there. So parathormone, thyrocalcitonin, prostaglandins, erythropoietins, these are the some of the hormones which are related to kidney functions. Okay. Now comes the homeostatic regulatory functions of kidneys. What are the different homeostatic functions? Okay, regulatory functions of the kidney. The homeostatic regulatory functions involves the process which basically maintains the state of homeostasis. Okay, so by continuously eliminating the what we can say the metabolic waste and the other impurities and even the surplus quantity of useful materials from the plasma, sorry, in the form of urine. So again and again, it is happening that we have to remove the waste. Okay, so it means we have to basically maintain the optimum requirements, optimum levels of everything in our blood. Okay, so kidney also operates certain other homeostatic regulatory mechanisms. Proper maintenance of the internal environment is known as homeostasis. Okay, like osmoregulation. Being the universal solvent, water is the universal solvent. Water is the actual vehicle in extracellular fluid to transport materials between the various parts of the body, and you know it that water is a universal solvent and it actually act as a vehicle. It actually acts as a solvent, okay, which helps to dissolve many extracellular fluid solutes to transport them. Okay, the volume of water in extracellular fluid tends to vary considerably due to several reasons, and because of many reasons, the level of fluctuation of water can be seen at the level of ECM, such as drinking, perspiration, diarrhea, vomiting. As described in previous pages, the kidneys maintain the water balance in extracellular fluid by diluting or concentrating the urine. Very important. As described in previous pages, the kidneys they maintain the level of water, they maintain the water balance in ECF by diluting or concentrated urine. Okay, so the level of water in extracellular fluid it is to be maintained, and kidneys are helping them out to maintain the water balance in extracellular fluid by diluting or concentrating the urine considerably. If it is, the urine is diluted, then we have to make it concentrated. If it is hypertonic or hyperconcentrated, then we have to dilute it. Regulation of osmotic pressure. So kidneys they help to maintain the osmotic. It helps to regulate the osmolarities. To regulate the osmolarities. Osmolarity of the cytoplasm mainly due to the proteins, especially proteins like albumin and the other colloidal proteins. Colloidal proteins and potassium and phosphate ions, where that of the ECF is mainly due to sodium, chloride, and bicarbonate ions. Very important. The osmotic pressure, the osmolarity of the cytoplasm in intracellular fluid is because of phosphate ions, potassium ions, and proteins, while that of extracellular fluid is because of sodium ions, chloride ions, and bicarbonate ions. In spite of there is a huge difference in the chemical composition of both the fluids. The intracellular cytoplasm and the extracellular, that is interstitial fluid, must be isotonic. Okay, because if ECF becomes hypotonic, cells will absorb water, then it will lead to edema, swell, retaining appropriate number, mainly of sodium and chloride ions. Kidneys maintain the normal osmolarity of extracellular fluid. Okay, so kidneys are concerned with maintaining the osmolarity of extracellular fluid. So if the ECF becomes hypotonic, the cells will absorb water and they get swelled up. Okay, so it helps to maintain the osmolarity of extracellular fluid. Regulation of pH. Concentration of hydrogen ions like NaH2PO4. Very important. Sodium hypophosphite in extracellular fluid is to be regulated. It act as a buffer in ECF. So it act as a buffer is to be regulated at a constant value, usually expressed as the pH. Okay, pH is basically the potence of hydrogen, that is power of hydrogen, which is mathematically or chemically equivalent to minus log of H plus. The normal pH of yeast is 7.4, that is equivalent to the pH of blood. At low pH, mean at the condition of acidosis, that is a high H plus concentration causes acidosis, while the high pH causes alkalosis. Both of these conditions severely affect the cellular metabolism because enzymes they are very much sensitive to pH. So if the pH levels are not up to the mark, if they are not appropriate, the enzymes are not going to work. They become non-functional. So several special control systems therefore operate in the body to prevent the conditions of acidosis and alkalosis. This process is compensatory mechanism. Okay. The system is called acid-base buffer system. Kidneys play a key role in maintenance and operation of these systems. Okay. Further, the kidneys regulate the hydrogen ion concentration in the extracellular fluid by excreting acidic or basic urine. Okay. So it helps to maintain, it helps to regulate the pH. Now, regulation of electrolyte concentrations in ECF. The kidneys regulate not only the total concentration of water, okay, and electrolytes in ECF, but also the concentration of individual electrolyte separately. Okay, like the sodium ions in extracellular fluid, if they are more around 135 milliequivalents and inside the cell they are less, so then this condition is to be maintained anyhow. Okay, this regulation is very complex, right? This regulation is very complex and is accomplished by the tubular reabsorption and secretion under the control of hypothalamic and adrenal hormones. Very important. So under the action of hypothalamic and adrenal hormones, this level of this electrolyte balance of ECF should be maintained anyhow. Okay, when we talk about that the composition of urine, so the composition of urine is basically having around 15 grams of NaCl per, you know, 15 grams of NaCl. Okay, 3.3 grams of potassium ion, zero point 0.3 grams of calcium ion, zero point 0.1 grams of magnesium ions. Okay, these are per deciliter. Okay, grams per deciliter. Okay, uric acid is zero point 0.7 grams per deciliter. Porphyrin is 50 to 300 micrograms per deciliter. Okay, creatinine 1.6 grams per deciliter. Creatine zero point 0.1 grams per deciliter. Urea 30 grams per deciliter. So, in this is, I mean, I am having the chart. Okay, with the help of chart, I am basically telling you that all these are optimum concentrations and they need to be maintained anyhow. Okay, so you don't have to learn all these concentrations. It's right. It's fair enough. You don't, learn them. Just help them to, make an image of it that okay, the concentrations need to be maintained. Now, the regulation of RBC count in blood, that is erythropoiesis in oxygen deficiency or hypoxia. The kidneys secrete an enzyme in the blood. This enzyme reacts with plasma globulin to form erythropoietin. This enzyme basically reacts with the globulin of plasma to form erythropoietin. The later substance stimulates the bone marrow to produce more RBC for enhancing O2 intake in the lungs. To form more and more amount of oxyhemoglobin, they need more amount of RBCs, they need more amount of hemoglobin. That is why it is important to produce more and more amount of, you know, RBCs, red blood corpuscles, if required. RAS system we have already done. Okay, so we are not going to, take it, consider it. Okay. Now, now we are going to talk about the urine, its composition. Okay. First, we will talk about the quantity. An adult man normally passes around 1 to 1.8 liters of urine in 24 hours. The volume of urine depends upon fluid intake. It depends upon the amount of fluid you are taking, the level of physical activity, what about, what different types of physical works, physical activities you are involved in, type of food taken, what type of food you are taking. Okay, environmental temperature. Okay, increases urine output. Okay, less fluid intake and profuse sweating due to heavy physical work and high temperature reduces urine output to maintain the level of water, to conserve the level of water in the salts. Because if water will go, then salt will also go. Certain substances such as tea, they all are diuretics. Coffee, alcohol increases the urine output, so they are diuretics. These are said to be diuretics. Sorry. Physical properties. Urine is a transparent yellowish fluid because of, but becomes turbid or cloudy on standing because of the release of ammonia. Its color depending upon its concentration. Its color is due to the pigment urochrome derived from the breakdown of hemoglobin. Okay. The heme will break into biliverdin and bilirubin, which will form the urochrome from the worn out RBCs, dead RBCs. Color of the urine is altered by certain materials such as beet, vitamin B complex, and certain drugs, diseases. It is hypertonic to blood plasma. Very important is hypertonic to blood plasma. Its specific gravity ranges from 1.001 to 1.035, being slightly higher than that of water. Its pH is 6. Okay. It depends upon the diet. High protein food, fruits increases the acidity, whereas vegetables increases the alkalinity. Urine has a characteristic unpleasant odor because of the liberation of ammonia gas, a nitrogen. If allowed to stand, urea is degraded by bacteria to form ammonia, which imparts a strong smell to urine. Okay, so basically, around the total volume is 1200 to 1500 or 1800 ml, per 24 hours, like 1 to 1.8 liters of urine 24 hours. Okay, the volume of urine depends upon various factors we have discussed. Okay, the physical properties that urine is transparent yellowish fluid but becomes turbid or cloudy on standing because of the liberation of ammonia. Its color depending on its concentration. Okay, its color is due to the pigment urochrome, which is derived from the breakdown of RBCs. Okay, The hemoglobin, the heme part which breaks into the biliverdin and bilirubin, this will further convert into the pigments, okay, like streptochrome and urochrome, okay, which imparts the color to stool and the urine respectively. It is hypertonic to blood plasma, mean it is having more osmolarity than blood plasma. The specific gravity is also more, okay. The pH is 6. It if the person is on more high protein diet, then more nitrogenous waste will be there, more urea will be excreted. Okay, fruits, if they are taking more fruits and proteins, the acidity will occur. Whereas vegetables increases alkalinity. Okay, chemical composition. Urine consists of water, organic and inorganic substances. Water alone forms about 95% of it. Other substances only 5%. The organic substances are mainly nitrogenous organic compounds like urea, uric acid, creatinine, hippuric acid. These are organic substances. Of these, urea is a principal amount of principal component of human urine. The non-nitrogenous organic compounds include vitamin C, oxalic acid, phenolic substances include ammonia, and mineral salts such as chlorides, sulfates, phosphates of sodium, potassium, calcium, and magnesium. Sodium chloride is the principal mineral salt of urine. Urine also contains some other substances such as pigments, very important, pigments, and drugs, and some epithelial cells, leukocytes, mucin, enzymes, and hormones. There are some, this is the chemical composition. Okay, urine consists of water, organic and inorganic substances. Okay, water alone make 95%. Okay, so the amount of water urine is, if it is around 1200 ml per hour, then the water will be 1140 ml. Okay, total amount of solids are 50 ml, 50 grams, sorry. Glucose, no glucose, 0. No proteins, no ketones. Okay, urea 30 grams. Okay, urea is 30 grams. Creatinine is 1.6 grams. Okay, so there is a chart. Okay, so in case of man, the water makes 95 to 96%. Urea makes 2%. Urea makes 2% of total. Uric acid 0.2%, ammonia 0.25%, creatinine 0.5%, hippuric acid 0.025%, and salt 1%. Okay. There are some abnormal substances which are also released sometimes. Okay. Presence of proteins, albumin, bile salts, bile pigments, ketone bodies, blood, pus cells, microbes, and more than a trace of glucose in the urine is pathological condition. So these conditions are not normal. Okay, so because all these substances are very of, they are of high threshold. The presence of glucose, protein, blood, ketone bodies, and pus in the urine is called glucosuria. If glucose is present, then it's glucosuria. If protein is there, then proteinuria. If ketone bodies are there, then ketonuria. Okay. If blood cells are there, then hematuria. If pus cells are there, then pyuria, respectively. Okay. So I'll write. No, slide. Yeah, I'll write here. These all are abnormal constituents. Okay. Or it can be glycosuria. Presence of glucose in urine, diabetes mellitus. Then presence of ketone bodies. There are two terms. Remember, one is ketonuria and other one is ketemia. Ketemia is presence of ketone bodies in blood, and ketonuria is ketone bodies in urine. It is also a complication of diabetes mellitus or maybe due to conditional starvation. Then albuminuria. The general term is proteinuria if significantly albumin isn't present in urine due to, very important, glomerular nephritis. Glomerulonephritis. Okay, in which the inflammation is present in glomerulus and nephron. Okay, then the other condition is hematuria. Presence of RBCs in urine, blood in urine. Okay, usually it is seen in trauma, urinary tract infection, UTI, or stones. The other one is hemoglobinuria. The presence of hemoglobin in urine due to hemolysis, that is rupture of RBCs. Okay, some other conditions are pyuria, presence of pus cells in urine, caused due to infection in kidney. Okay, just a minute. So pus cells in urine is the condition for pyuria. And then there is an important term, xanthiniuria. Xanthine, presence of xanthine in urine. Xanthine is an intermediate which is formed during purine metabolism. During purine metabolism. Okay, it is caused due to the deficiency of the enzyme xanthine oxidase. xanthine oxidase. Okay, it can lead to formation of stones known as xanthine calculi. Okay, these are some abnormal constituents of urine which can be seen. Now, I will be talking about very importantly about something about urinary bladder, which being the part of the question every time. Okay, every time the histology of especially the urinary bladder is the part of the question paper. Okay, urinary bladder, very important. The muscular layer which is present, which helps in the voiding of urine, detrusor muscle. Okay, so they have three layers muscles. One is the longitudinal. In between, they have circular. Then the outer one is also longitudinal. The outer, middle, and the inner. Understood? Pudendal nerve. It acts on external sphincter. External sphincter. Okay. Around 700 to 800 ml of urine can be stored at a time. Okay. The process of voiding of urine is micturition. So, ladies and gentlemen, we will be starting the micturition and the nervous control of the micturition in the coming class. That lecture will be the last lecture, probably, of excretory system. We will try to cover different other accessory excretory organs along with the process of osmoregulation, osmoconformation. Okay, So till then, bye bye. Take care. Stay safe. Stay healthy.